present our keynote speaker, Dr. Helen Tai, a research scientist with Agriculture and Agri-Food Canada, joining us today from Fredericton, New Brunswick. Take it away, Dr. Helen Tai.
Hi, I'd first of all like to thank the organizers for the wonderful job that they're doing putting this Potato Summit together and for this invitation to speak to you today. I am here at the Fredericton Research and Development Center with Agriculture and Agri-Food Canada. I'd like to start with this land acknowledgement. This presentation is being delivered from traditional unceded territory of the Wolastoqiyik Indigenous people of Canada. And also a crop acknowledgement. Potato was introduced to the world by the indigenous peoples of Peru and Bolivia, whose ancestors began cultivation between 8,000 and 5,000 BC. Today, potato is the third largest crop consumed by humans, and you can see this beautiful picture of Lake Titicaca straddling the border between Peru and Bolivia. It is the site of origin of potato. I don't think I need to tell this group that potato is an important crop in Canada. It's the 5th largest agricultural crop and the largest vegetable crop. What I'd really like to focus my talk on today is why potato variety development needs innovation and to discuss some of those innovations that have come. So climate change is a big issue for agricultural producers. Here we see a map of the change in precipitation between 2051 and 2080 compared to 1975 and 2005 time periods. And you can see the one on the right is the spring map, and the one on the summer, the one on the left is the map for the summer. So climate change will bring wetter springs across Canada and hotter, drier summers to Canada, mostly in the Western and Central Canadian regions. So this is an issue for potato. Potato is very sensitive to heat and drought conditions under global warming. Heat will inhibit tuberization, and drought has a severe negative impact on yield. So these are issues that need to be addressed, and variety development is certainly one of the ways to do that. Potato is also susceptible to a wide range of pathogens and pests, leaving a large environmental footprint, as well as costs and risk of crop losses for the grower. And potato production is nitrogen dependent. So it requires nitrogen fertilizer, and nitrates interact with the soil microbes to produce nitrous oxide, which is a very powerful greenhouse gas. It's even more powerful than carbon dioxide. So mitigating usage of nitrogen fertilizer can be a way to mitigate climate change. And also, very importantly, variety development targets industry needs. So high quality potato products and development of new products needs varieties with appropriate tuber quality traits. And we should also note that low tuber quality leads to increased waste, and increased waste is really also wasted greenhouse gas emissions. So it would be very important to maintain that tuber quality and improve it so that we can reduce this waste in the potato production system and industry processing. Reducing costs and risks faced by growers is also a priority, and good agronomic traits are needed here. And in the whole system, all of the traits that are needed by the various parts of the industry, that all needs to be communicated to the breeder. So a good communication channel is needed to make the breeding programs relevant to what the industry needs are. So as you can see, there's multiple issues facing the potato industry that translate into a multitude of traits for the potato breeder to evaluate. And I would ask if you'd like to enter into the chat the potato traits that you're interested in and that you'd like to see improved. It'd be great to hear from you. Here are some of the potato traits that I've noted that are of interest for breeding. You can see there's a lot of them. Can't talk about them all today, but I think it's important to note that we address variety improvement through a breeding system. And in the AAFC potato breeding system, there are 400 parents and they are cross-fertilized. This means that the pollen from one parent is placed on the stigma of another parent, and sexual reproduction occurs. So this is your X-rated part of the talk for the day. Fruit is formed. This fruit contains seeds, and each of these seeds is a genetic individual. So kind of like you're genetically different from your sister and your brother, these seeds are all genetically distinct as well. They then get planted into the greenhouse, and 60,000 seedlings are planted a year. These seedlings are grown to form mini tubers. These mini tubers are then planted in the field. And 1 to 10% of those plants are screened and selected using visual selection by the breeder. And this is followed by another 5 to 6 years of multi-hill trials. And then those trials, towards the end, those candidates would be screened for disease and pest resistance traits. Throughout this 6 years, there's fry testing. So there's a lot of selection that occurs through the system. In the end, only about 0.02% of that 60,000 makes it to a national evaluation trial. And there, only 0.02% of that 60,000 makes it to a commercial release. And then after that, there's a seed increase that's required. One tissue culture plantlet is provided for plant propagation into nuclear seed, and elite seed production occurs to provide commercial level seed for commercial production. Then there's also a process of on-farm adoption that also can take quite a while as well. So basically, it takes time to develop a potato variety and to get it adopted. And so, as Erica will tell you more in her talk, and also addressing this in my talk, we need better varieties faster. So we need a better way to get to improved potatoes. So breeders need to find balance. So there's what's desirable, which, you know, I put up a lot of the desirable traits in the talk, but also what's really important is what is feasible. And oftentimes what is feasible is the most important thing because it's what the breeder can do. So how can we make variety improvement and all the traits to be screened feasible, and more feasible actually? Let's look at how potato compares to the other major crops: maize, wheat, rice, and potato. As you can see, the yield for potato is quite good, but the yield increase over this 50-year period from 1966 to 2016 for potato lags behind maize, wheat, and rice. And this is because potato, in part because potato is very complicated genetically. It's an autotetraploid crop, and the other crops, maize and rice, are diploids. But wheat is a hexaploid, but it's an allohexaploid, meaning that it's more like 3 diploids, so it acts like a diploid. So this schematic diagram is meant to show you how much more complicated autotetraploid genetics are. And you can see these are red and blue lines. Just sort of note how many more red and blue lines there are for a tetraploid potato than there are for the diploid. And this gives you an idea of the complexity that is present for the breeder in dealing with potato. So this means that some genes, bad genes, can get hidden. The good genes have a hard time getting expressed. So potato breeding needs innovations. And what I'm going to talk about next is how the potato breeding system can be improved. One of the developments that has happened is the development in new DNA sequencing technologies. And this is a diagram produced by the NIH showing the cost per human genome. Of course, it's the same for all genomes, and there's this dramatic drop in costs. So something that cost $100 million in 2001 only costs $1,000 now. So this sequencing this genome sequencing has dropped a lot in cost. And this provides opportunities. And the opportunities would be to find genes and to develop markers for selection in breeding. So what's happened with potato? The Potato Genome Sequencing Consortium pooled international resources to get a potato genome sequence in 2011. And David DeCoyer, our breeder, and Shuqing Li from AAFC, they didn't directly participate in the sequencing consortium, but provided resources that the consortium did use, and this consortium was very successful, and they sequenced a doubled monoploid potato. This doubled monoploid potato is a simple potato in that it only has one genome instead of 4. And this was a laboratory generated plant that came from an anther culture. And you can see it here. DM is the doubled monoploid, and you can see that it's very small. It also produces very strange looking tubers, so it doesn't really look like a commercial potato variety, but it was very simple, and simplicity was needed at this point in time in order to get a genome sequence. So this doubled monoploid has been the reference genome sequence that has been used by the potato community since 2011. More recently, in fact, last month, another international effort put together the genomes of 6 tetraploid commercial potato varieties. I'm very happy to say that myself and Kyle Gardner from AAFC were part of this sequencing effort. Atlantic, Altus, Avenger, Castle Russet, Columba, and Splunta are the varieties that were sequenced. You can see that they cover a range of different market types, and 4 of them are shown below. So you can see that they are looking very much like what we would expect of a potato. This effort was led by Richard Finkers from the Netherlands, and Genevieve Hoops from Michigan State University did most of the bioinformatics work on this project, and she was a student in Robin Buell's lab. So potato genome sequences advance discovery of genes and DNA markers and applications in selection and breeding. And the genomes are large. So the monoploid potato genome is 850 million bases. The tetraploid genomes are over 2 billion bases. So there is need for high-level computation, and this comes from combining computer technology with DNA sequencing in something that we call bioinformatics. And I like this little cartoon here where it says, "I hate being a DNA molecule. There's so much to remember." And this is truer now than ever before. So what can we do with this genome sequence? Well, we'd like to develop something called marker-assisted selection. Marker-assisted selection uses genetic mapping to identify markers in the genome. So this is like having a road with signs, and the signs help to mark where you are on the road. So you'll know that you're near the house that you're going to. And so the house is our gene. So quite often the markers are not the gene, they're near the gene. And sometimes they are the gene, but quite often the markers are near the gene. So we need to make a map or a genetic map of where all the markers are and see where they are relative to each other. Then we collect a whole lot of trait data, and we put the marker and the trait data together, and we associate the traits with the markers. And in this statistical association, those markers, which are bits of DNA sequence, they can then be used for markers in selection and breeding. So you do a genetic mapping exercise, find your markers, and then use them in selection and breeding. And this is really good for single gene traits, such as our genes for disease resistance. Another approach is genomic selection. In genomic selection, genetic mapping is not required. Instead, a training population is used to develop a prediction model, and this is good for complex traits such as specific gravity. So in genomic selection, there's a training population, and then genetic marker data is collected for thousands of sites. So you really need a really, really high level of marker density, higher than you would need for genetic mapping. The trait data is also collected, and the genetic marker and the trait data together are fed into an algorithm to produce a genomic prediction model. And so I often like to say that genomic selection is kind of like online dating. In online dating, you provide all sorts of data about yourself through the questions, and then that is used in these online dating algorithms to figure out which are the two individuals that would make a good couple. And we're kind of doing the same thing. We're figuring out which two potato clones can be crossed together to give us a good progeny. And what the genomic prediction modeling will output for us are genomic estimated breeding values, which use this genetic marker data to predict what the trait would be, and also helps us to determine what a good cross would be. Imaging and sensing technologies have also really come a long way, and Erica will talk more about this in her presentation. We are also using IT technologies to integrate large amounts of trait and genetic data. And PotatoBase is a web tool that's developed by the Boyce-Thompson Institute by Lucas Mueller. And this is allowing us to put in there all of that marker data as well as all the trait data. With all this data that's being collected, you really do need IT technologies as well to sort it all out. So what does a marker assisted selection and genomic estimated breeding value tetraploid breeding scheme look like? Well, the parents can be selected using mass and GEBV. The parents, currently you use historical data, specific and general combining ability information to select the parents. You add on to that marker data and genomic prediction data, and then you make your crosses. Again, you'd have your F1, which are our progeny. This is the genetic terminology used to describe progeny or used to define progeny, and you'd have about 60,000 of them. You do the visual selection, and then you go to these multi-year evaluation trials, and selection can then be applied again at this point to select out individuals. So you might select more than your 1 to 10% if you're planning on using additional marker and genomic prediction selection. So the marker assisted selection and the genomic prediction using genomic estimated breeding values can also be used to increase intensity of selection. So you can select parents, and then these parents can be grown in the greenhouse. And in the greenhouse, these seedlings can then be tested for marker and genomic data to select another set of parents. And so a second crossing occurs in the first year. And then this second cross produces the F1 clones that go into the field for additional multi-year evaluation trials and additional marker and genomic prediction selection. So there's various different ways to use markers and genomic estimated breeding values. And this is something that we're researching right now in one of our projects. So let's come back to this slide comparing potato with maize, wheat, and rice. Another aspect of breeding for maize, wheat, and rice is hybrid vigor, especially for maize. So diploid F1 hybrid breeding is the breeding system for maize. And inbred lines are used for breeding. And the advantages of this system is something called heterosis or hybrid vigor. So you can see there are two parental lines, inbred A and inbred B, and they are crossed together to produce this hybrid. And the performance of the hybrid far exceeds what the performance of either of the parents are. So crossing these two inbred lines produce something called hybrid vigor. And this hybrid vigor is due to something that we call heterozygosity, meaning that the two inbred lines had relatively homogeneous or homozygous genetic content, but when they get cross together in the progeny, this becomes more mixed and heterozygous. And this heterozygosity is thought to underlie the hybrid vigor. The diploid F1 hybrid breeding also allows for more effective use of marker-assisted selection to increase genetic gain, and true seeds are used for propagation and commercial production. So going back to this diagram of diploids and tetraploids and looking at our very complicated autotetraploid potato, the question always comes up, how about breeding potato as a diploid crop? Well, diploid potato is already used for cultivation and breeding. In fact, indigenous peoples in South America and North America have cultivated several diploid species, and they're usually called land races, that produce tubers in addition to Solanum tuberosum, which is our cultivated species that we use here in North America. There are over 100 cultivated and wild species that produce tubers. Most are diploids. Some species can induce tetraploids to become diploids. And diploids have been developed at AAFC. In fact, a variety was released, AAC candy cane. The thing with diploids is that thus far they've been noted to have decreased yield. So additional improvements are needed for diploids to overtake tetraploids. Nevertheless, diploid F1 hybrid breeding has been developing under several breeding programs. And what diploid F1 hybrid breeding will involve for potato, as I said before, is the development of these inbred lines. So you have to have selfing diploids, which is a critical thing. So you need to find a source for selfing. And then those lines need to be selfed for 6 generations to get inbred lines. Once you have the inbred lines, you create these F1 clones, which are then screened in multi-year trials, not unlike what happens on this left side with the tetraploid breeding. And then once you've identified a good clone, instead of kind of advancing that along to seed production as you would in the tetraploid breeding system, you would then identify the parents that gave rise to this very advantageous clone. And then you would do a large-scale crossing. So a large-scale pollination would happen. And then you'd produce true potato seed. And then you'd go into a true potato seed-based seed tuber production. And it's not entirely clear at this time how that seed tuber production system would work for a diploid F1 hybrid breeding. Would it just be true potato seeds? Would it be hardy seedlings like tomato? Would it be a combination of hardy seedlings and perhaps seed tubers again? So this is still something that needs more investigation. So I'd also like to address where breeders can find new genes for improving traits. So wild relatives are a source of beneficial traits. And as I said before, there's over 100 wild relatives of potato that can cross with the domesticated potato, Solanum tuberosum. On the left, we're looking at 6 wild species that have been investigated by me and our entomologist, Duval and Pelletier, for Colorado potato beetle resistance. But the wild species also have late blight resistance and resistances to a lot of other pathogens and pests, as well as resistance to heat and drought and a lot of the other abiotic stresses that we're concerned about. And you can see here in this picture from the International Potato Center, the great diversity in tuber types that you can also get from the collection of wild potatoes as well as land races. The use of DNA markers is really helpful when you're doing intercrossing with wild species. And these kinds of intercrossing, we call this germplasm enhancement breeding. In these intercrosses, what happens is you bring over this beneficial gene from the wild species, but then the wild species doesn't often make good tubers. It doesn't often have good adaptation to our climate. And so you do have to do a lot of back crossing. And this is a development of a breeding line by David DeCoyer and Yvonne Peltier. They made this cross between this wild species Brevicali and Solanum tuberosum. And then there was a series of back crosses from the progeny back to the Solanum tuberosum. And so this is quite a laborious process. And this generates another breeding line that goes into the breeding system, which then outputs the varieties. So all the breeding methods discussed thus far utilize natural variation from existing potato lines in wild species. Another way to introduce beneficial traits is through gene transformation. And AAFC does not use gene transformation for variety development. AAFC uses natural variation breeding using crosses, as I indicated before. So what is gene transformation? In gene transformation, there's a circular piece of DNA called a vector. This vector is then introduced into a bacteria called agrobacterium. The agrobacterium then infects plant tissues, and the vector has an antibiotic resistance gene so that you can put these plant tissues into tissue culture media containing antibiotics to select for the plants that carry the foreign DNA. Then that is grown in tissue culture and then into the greenhouse. And then there's a molecular screening for the transformed plants. This can be done by gel electrophoresis, as is shown here, or it can also be done using PCR. These plants are grown in the greenhouse and then put out into the field. You do need some licensing from CFIA for that to happen, and I'll discuss that. And this leads to the development of plants with improved traits. In addition to agrobacterium, you can use gold tungsten particle coating of your DNA and a gene gun to shoot the DNA into the plant tissue, and then the same process occurs. So as I said, gene transformation involves genetic engineering of a vector in the lab to carry a new gene into the plant. And the crops developed using this technology are referred to as genetically modified. So in these plants that contain new genes, there's subcategorizations. So transgenics involve insertion of genetic elements from sexually incompatible organisms. For example, putting a bacterial gene into plants, which is like the BT gene for insect resistance is placed into a plant. Cisgenics involves insertion of a genetic element from a sexually compatible organism. So this would be like putting a potato gene into a potato. So for example, using the vacuolar invertase RNAi, the antisense of it, to turn off vacuolar invertase would be a cisgenic. Gene editing is a newer technology, and gene editing involves genetic engineering of a vector to carry a gene editing molecular machine into the plant. So the gene editing machine is a guide RNA targeting a plant gene together with a nuclease that cuts the DNA. And the crops developed using this technology are referred to as gene edited or GE. So the gene editor is, in this case, I'm talking about a CRISPR-Cas9 system. Now, you may have heard of CRISPR-Cas9. Jennifer Doudna and Emmanuel Carpentier won the 2020 Nobel Prize for discovery of this CRISPR-Cas9 system. The gene editor is this thing called a Cas9 nuclease. When that gets transformed, it gets expressed in the plant to get transcribed into an RNA, and then that gets translated into this Cas9 nuclease protein. The plant gene, on the other hand, it gets transcribed into an RNA, but it does not get translated into a protein. It stays as an RNA molecule. And the RNA molecule has this hairpin loop, and that allows it to combine with the Cas9 nuclease to form this CRISPR-Cas9 complex. In the CRISPR-Cas9 complex, this single guide RNA that is within it guides it to the endogenous plant gene. When it reaches the endogenous plant gene, these molecular scissors cut the DNA and a mutation is induced. You can also put in there another piece of DNA that you'd like to insert into this cut piece of DNA. So there's a lot of flexibility in the system, and a lot of molecular biologists are working to make the system more accurate and more efficient. The other thing is that the gene editing machinery can be removed. So then all you're left with at the end is just this gene edited DNA without any foreign DNA at all. And this is also a point where you can go back and use natural variation and intercrosses to get rid of some of this gene editing machinery. And there's a variety of other methods that can be used. And also, there is technology now where you don't have to do the DNA transformation. You can just use different methods to introduce the guide RNA and the nuclease into the plant and get this DNA editing to occur without ever introducing any foreign DNA. So how are genetically transformed plants regulated? In most countries, the regulation of technology is based on the method of gene transformation use. So this would be like a difference between a transgenic and a cisgenic. They'd be regulated differently and they'd be regulated differently again from gene editing. In Canada, however, the regulatory framework is called plants with novel traits. Plants with new traits generated using gene transformation are regulated similarly to plants with new traits generated using other technologies such as cross-pollinations. So there's no distinction between how a plant with a novel trait was generated. Plants with novel traits are assessed for their potential to become a weed or invasive of natural habitats, potential for gene flow to wild relatives, potential for a plant to become a plant pest, and potential of a plant or its gene products to negatively impact non-target species. And of course, there's the potential for impact on biodiversity. So all of these are part of the assessment, and then only then would you get a permit to put a plant with a novel trait into the ground. So there's a lot of regulation of this technology. There's also the Cartagena Protocol, which is an international agreement that aims to ensure safe handling, transport, and use of GMOs. In spite of all of this regulation and so forth, there's still a lot of apprehension out there about genetic modification and gene transformation and transgenics and so forth. And this is part of the reason why we don't develop we don't use these technologies in developing varieties at AAFC. So if we look at the history of what's happened with potato, Monsanto commercialized a transgenic potato variety New Leaf in the mid-1990s, which expressed a bacterial gene Bt, conferring insect resistance. Anti-GMO food protests led to removal of New Leaf from the market in 2002. So the situation demonstrates that consumer acceptance will be required for commercialization of varieties using gene transformation technologies, and that's something that needs to be kept in mind. If we look at crops where GM has been adopted and commercial varieties have been in production, this would be GM soybeans, corn, cotton, and canola. They have a global, there's a recent study that shows the global net economic benefits at the farm level were $225 billion for the period between 1996 and 2018. Over that same period, insect-resistant GM crops resulted in global reduction of insecticides by 8.3% and reduced carbon dioxide emissions equivalent to removing more than 15 million cars from the roads. So you can always find out which crops have been approved for commercial production with the International Service for Acquisition of Agri-Biotech Applications. The website is here. However, as I said before, approval by a regulatory body, and in Canada, it's the Canadian Food Inspection Agency, is not the last step for getting your GM crop commercialized. You still need to consider variety adoption. And of course, as I said before, the consumer is very important in this process as well. But just a final word here about variety adoption. As I said before, a lot of different players are involved in the industry, and all of these parts of the industry have needs and have issues that need to be addressed in the variety development. And so all of this information needs to feed back to the breeder in order for successful variety development to happen. And I'm just going to show you the top 15 registered potato varieties in Canada. And you can see that Russet Burbank still really sits very much at the top of this list. This is a over 100-year-old variety. And so it would be, this variety also doesn't have very good climate change adaptation, susceptible to a lot of things. So it would be good to see if we can do better with variety development and variety adoption. So just to summarize, I talked initially about the need for developing varieties, the development of new DNA sequencing technologies that reduce cost for these technologies, and it's bringing innovation in developing marker-assisted selection and genomic selection. And also there are new breeding schemes that are being developed using markers and genomic selection, but also with diploid F1 hybrid breeding. And wild relatives are a source for new genes, as well as gene transformation and gene editing. And of course, variety adoption is key for the industry. So I'd like to end by saying that I'm the president of the Potato Association of America this year. And I'd like to ask if you could visit the website. And if you are not already a member of this organization, please consider joining. It's a wonderful place to network with other people in the industry and to connect with potato scientists like myself and some of the others on this at this summit today. And I'd also like to announce that the Potato Association of America is going to be having our 106th annual meeting from July 17th to July 22nd at the Holiday Inn downtown in Missoula, Montana. This is going to be an in-person conference, but there are going to be virtual options in case you're not able to go in person. So please contact Erin McLaughlin, our conference organizer. So thank you very much, and I'd be happy to take questions.
Well, thank you so much, Dr. Tai. That was fascinating research that you're sharing. We do have some questions coming in here, and just some comments, even just to thank you for providing an excellent overview and presentation. So a lot of this is really complicated research, of course, but I do want to start by just going back to what you had mentioned in the beginning, asking our audience to share some of the traits that they like to see most. So I'll remind the audience to do that and share those in the comments with Dr. Tai. But Dr. Tai, what traits do you find to be most exciting when it comes to potato breeding? What are you looking for? What really gets you excited about some of the research that you're doing?
Yeah, that's a really good question, you know, and I think that I've done a lot of work on Colorado potato beetle, and that's definitely very, very interesting to see how the beetle interacts with the plants and what the beetle likes, what the beetle doesn't like, and the different strategies that the plants use to fend off the Colorado potato beetle. But as I do more research in this area, I'm finding that you can't just, with the Colorado potato beetle, it'll be more than just varieties. You'll also need to have integrated pest management. And that's important to consider as well if you're trying to use varieties. You know, consider also using IPM strategies together. And the other traits, of course, are disease resistance, and those are also very interesting traits. A lot of developments there with discovery of resistance genes and things like that. And I've also worked on nitrogen use efficiency and understanding how a plant responds to nitrogen. And of course, drought and all of them. So yeah, I guess I don't really have a favorite trait, Stephanie. They're all interesting to me.
That's a good answer. And you know, there's some comments here in the chat too. If folks are looking at, you know, things like drought and fungal resistance, as you say, from year to year, those topics are going to be varying in what growers are most interested in, of course, but great to have some information from folks like you to share. So I know you mentioned a little bit about the timeline of release and all of the regulatory kind of steps that are necessary to take to get your research into market. Is there anything in the immediate future that you can kind of hint at or share for growers who are wondering, when might these, potatoes with drought tolerant traits or other really exciting breeding traits and breeding developments that you've been working on, when are those going to be coming to market? And is there anything that you can kind of hint at for the next couple of years?
I'm gonna let Erica answer some of those questions. She's got an excellent presentation, and I think that Erica, she's on the breeding team, and she can really address these issues a lot more. And
Wonderful.
all I can say is that in addition to all the gene sequencing technologies that are out there, we are looking at a lot of other kinds of things, sensor technologies and use of. And today I went to a really exciting talk on ground penetrating radar. So these sensing technologies are also going to really advance things for breeding, and using those in combination with the genomics technologies is really going to allow us to do a lot more. And so I think that I'll let Erica talk about some of this. The breeding program isn't, and as I said, the other thing I want to say is that it's not just one trait. The breeders are often dealing with groups of traits at the same time. So yeah, drought is a very important issue. I think that this is something that we'll probably need to pay attention to a lot more as we go forward.
Definitely. We've got time for just a couple more questions, and there are a few really good ones coming in. Now, you addressed consumer demands and a little bit of the consumer kind of backlash against some of these more intensive genetically modified traits. And arguably, consumer demands are one of the biggest drivers for varietal development and acceptance. So we've got a question here noting that and asking, from your perspective, how would you recommend that we could better educate consumers on the benefits of this? Because as much as we'd love to share a presentation like yours to the entire world, it's not possible to do that all the time. So is there anything that growers can do to help educate consumers and the industry as well?
Yeah, I think that one of the issues that I think is that we talk about genetic modification like it was one thing. And I hope that my talk today will illustrate to you that it's many different things. And so, I had one of my mentors at AAFC, Bernie Seabarth, used to always like to start off an answer to the question with "it depends." and I'm going to take a lesson from Bernie today and say it depends. And that's what you have to tell people when they say, I don't like GM foods. And then you say, well, it depends on what they're being used for. Mm-hmm. In some cases, yeah, maybe it isn't such a good idea, but in other cases, it might be the only option. And so we can't look at them, lumping them together. We have to look at each one case by case. So when somebody says they don't like GM foods, you can also always say, why? What is it? What's the property? Because they're not all the same.
So true. And I think there's still a lot of misunderstanding about that factor right there. Genetically modified doesn't mean one thing. So that's a great way to start that conversation for sure. I think we've got time for just one or two more questions. So there's one here in the Q&A box that says, with the changes to breeding and the future with true potato seed, are there considerations that need to be addressed when considering the future needs of the seed potato certification system?
Yeah, I think that this is something that we're all sort of wondering about with diploid potato. The seed certification system will have to make adjustments to a system that uses true potato seed as opposed to, seed tubers. There will be differences there. If there are sort of discussions where the true potato seed is then, you can germinate it in vitro, and then you're going back to the usual plant propagation system, tissue culture plant propagation system to get to elite seed. So there is those possibilities. But the advantage of using true potato seed is the fact that you can make thousands and thousands of seeds with these crosses. Some of the lessons learned from tomato would be probably a good place to look to see some of the disease issues that they face with hybrid seed production in tomato. They're different from ones that we might face with seed tuber production. So there's still a lot to be investigated, a lot to be learned from some of our other Solanaceae crops that are produced using hybrid seed.
Okay, I hope that answers your question. And there's another question here in the chat about wondering if you could address breeding techniques to reduce the impacts of nematodes.
Yes, nematode resistance. I've seen some work done. It's interesting. Nematode resistance also can be in the plant. It's genetically controlled by our genes, but different R genes. So nematode resistance is always a little bit difficult to study because it's not always easy to grow nematodes in the lab and to to have them infest a plant, and quite often it's a root infestation. So this nematode resistance is really important, but very, very difficult for geneticists and breeders to get a hold on to. And also sometimes it's better just to have it happen in the field, and to have somebody constantly maintain a nematode infected field is also not a good idea. So nematodes are very challenging. I think that the way to approach nematodes may be to look more at what's going on in the plant community overall, and to look at it from a molecular perspective, what genes have been effective in other organisms and other crops, and see if we can find those equivalent genes or homologous genes, as we would call them, in potato.